Don't need your.
It's been a great pleasure to introduce Dr. Claudia Kawas. Dr. Kawas holds an endowed chair in clinical neuroscience at the University of California, Irvine. She's professor of neurology in the School of Medicine and professor of neurobiology and behavior in the School of Biological Sciences. Her primary interests are aging and dementia. She has published over 100 articles in peer-reviewed journals in these areas. Dr. Kawas is a graduate of Swarthmore College, completed her medical studies at the University of Louisville, then her neurology residency training and fellowship in dementia and aging at Albert Einstein College of Medicine in the Bronx. After 15 years on the faculty at Johns Hopkins University, Dr. Kawas moved to the University of California, Irvine in 2000. She is principal investigator of a unique study, the 90+ study, and associate director of the UCI Institute for Memory Impairment and Neurological Disorders. She will talk to us today about some of the results coming out of the 90+ study. It is one of the largest studies of the oldest old in the world, with more than 1,400 people enrolled. Please welcome Dr. Kawas.
I really can't tell you how thrilled I am to be here. I had the pleasure of visiting Orcas more than 20 years ago for the first and only time, and it feels very, very, very special to be back. And you live in a beautiful place. So thank you for letting me share it, and I'm also looking forward to sharing with you what a lot of 90-year-olds have taught me. So before I get too far along, though, I have met a few of you, and I know that there are a couple of potential subjects in this room. So anyone who's willing to fess up, who's over the age of 90 in the audience? Raise your hand. And one of the things that you're going to see later on is that the fact that you're in this room listening to this lecture actually is associated with a longer life. So it's a pleasure. Who knows who this lady is? Yeah, a lot of people I can tell. Madame Jean-Louis Calmet, and she was born in Arles, France. She, as a child, sold paintbrushes to Van Gogh. Yeah. And many years later, in fact, she was on a public television show about Van Gogh where she described him as, well, I don't think she liked him too much. He was dirty and disgusting, and she thought his artwork wasn't too good either. But my favorite story about Madame Calmet is that when she was 90, she made a deal with a 47-year-old man for her apartment in Paris. And it was a very desirable apartment, and it's a very common thing to do there, that this 47-year-old man would pay her a monthly fee. And she continued to live in her apartment, but when she died, he would get it. And if he paid every single month for 10 years, he would pay the market value of the apartment at the time. So she's 90, he's 47, sounds like a pretty good deal. And in fact, he did pay for 10 years. And then he paid for 20 years. And then he paid for 30 years. And then he died. And his widow paid for 3 more years because Madame Calmet lived to be 122 and a half. And she's in the Guinness Book of World Records for the longest documented living human being. So, why do we want to study someone like Jean-Louis? And that's what I'm going to talk about today. I mean, first I'm going to talk a little bit about oldest old, which I'm defining as people over the age of 90. Although in previous decades it was considered to be people over the age of 85, but that's passé now. A census report last year said that those over 90 comprise the oldest old demographically and are their own kind of group, and we really need to know more about them. So we were kind of prescient in the 90-plus study, which we started in 2003. And the results I'm going to show you are some of the things we've learned about risk factors or protective factors for longevity, that is how long people live, risk factors and incidence of dementia, which, as most of you know, is things like Alzheimer's disease or other things that interfere with our thinking. I'm going to show you a little pathology and a little radiology of individuals that we've been learning in the 90-plus study, and then I'm going to try to come to some kind of conclusions. So, right now in the United States, there's only about 2 million people over the age of 90, but by the middle of the century, this is going to happen. We will have a quadrupling of the number of people in this age group, actually worldwide, because throughout the world, people over age 90 are the fastest growing segment of the population. Now, so when we first started this study, actually, there was very few lines after that dotted line. Let's see if I can take it over there. Because people, for the most part, hadn't studied individuals over age 90. But what you see here is different studies are represented by the different colors. And what they show is, by age, your chances of getting dementia in a year. So you can see down around age 75, all the different studies have more or less the same number. And you can also see that between ages 65 and at least 85, that the risk of developing dementia doubles, actually, with every 5 years of life. But after age 90, when we first started the study, there were virtually no lines over here. Now you can see 10 years later, there's a lot of lines there. But in fact, you can see that they give you completely different estimates. So some studies have suggested that if you're 95, your chances of getting dementia are actually only about 3%. Some studies have suggested that if you're 95, your chance of getting dementia is about 35% per year, and everything in between. And you can see the difference between 3% of 10 million and 35% of 10 million is a really big difference in the number of people who would have dementias like Alzheimer's disease. So from a public health standpoint, it's really crucial for us to figure out which of these is correct. And that was one of the goals, in fact, of starting the 90-plus study. Because when we started the study, we realized that we really didn't know what the risk of developing dementia was. And in addition, we also didn't realize what are the causes of dementia when you become over age 90. So here in 2000 was a study done by one of my colleagues, and looking at a large number of subjects who died with dementia, he said, well, in your 70s, 95% of the time, the pathologist can say this is the cause. Either it's from Alzheimer's disease, it's from strokes, it's from mad cow, it's from whatever it is. But as people get older, about 5% in your 70s, the pathologist would say, I'm really not sure what the problem is. But by the time people are in their 90s and die with dementia, 1 out of every 2 cases, the pathologist would say, I'm not sure what's causing the problem. This looks like a normal brain to me. So we don't even know what causes dementia in people who are over the age of 90. We don't know the risk factors for those dementias. We don't know the protective factors. We don't really know what makes it so that some people live to be 90, 100, or even 122. And we don't know the risk factors related to the kinds of dementia that they get. So we wanted to learn some of these things, and so we started the 90-plus study. And what I'm about to show you actually is a little videotape. It came from it's a 5-minute videotape that's an excerpt from a public television show that was made on the study in 2004. And it shows a couple of our participants and things, and I think you'll find it really nice. And I especially want to thank the gentleman who figured out how to show this videotape.
Unlocking the mysteries of Alzheimer's. It's a major challenge as America grows older. In fact, people over 100 are one of the fastest growing segments of our population. But we found a unique study on aging that's happening right in our own backyard. Philip Bruce takes us to the source in Orange County. It's the community of Leisure World in Laguna Woods, where doctors have a rare window into the aging process.
Nice to meet you.
Well, thank
Would you
you.
like to come on back and get started?
She was born 2 years before the Wright brothers took that first flight at Kitty Hawk. But after more than a century on this earth, Viola Cron's body, and most of all her mind, remained sound.
Looks like mushroom.
Perfect.
A camel.
Good.
And this is one of the ways we can tell just how sound Viola really is. She's being tested on how well she can identify familiar shapes and put them into words.
We have very few people who can do that.
Really?
Yes, and you did it.
The exam is being carried out by the folks at UC Irvine's Institute for Brain Aging, and Viola is an old friend here. They've been tracking her and a few thousand other seniors from Orange County for the past 20 years, trying to find out what makes them tick.
We're finished. You are terrific. Thank you very much.
And don't you forget it.
Viola hasn't forgotten much, and her memories are enough to fill a movie. In the 20s, she was a budding Olympic diver after being discovered among a bevy of swimmers at the old Los Angeles Athletic Club.
The coach, friend Katie, started me diving, and I never went back to swimming. I became a diver, and then I married my coach, friend Katie. And of course, after that, then I simmered down and didn't compete anymore.
She never got to swim for a medal, but on her 100th birthday, Viola did get to swim for Jay Leno on NBC's Tonight Show. And there on national TV, she redefined what it means to be an active senior.
Anytime you want, baby, go ahead.
2 years after that dive, Viola admits she isn't always as able on dry land as she is in the pool. As I discovered, she hasn't lost her sense of humor. You're 102, can you believe that?
Can you?
Remember we told you that UC Irvine's doctors have been tracking Viola for the past 20 years? Well, it's not because of her adventurous past. It's because of where she lives. You see, Viola is a resident of Leisure World, a retirement community of some 18,000 souls near Laguna Beach. Ever since 1981, thousands of seniors there have been taking part in a yearly voluntary survey. They answer questions about what they eat, what they drink, how much they exercise, even what they think. And as Leisure World's population gets older, researchers compare the answers to how each individual is aging. It's especially valuable in studying how the brain ages, and determining if such things as lifestyle, diet, and mental output have anything to do with who gets Alzheimer's. Dr. Claudia Kawas, one of UCI's top neurologists, has taken the study a step further. She's bringing in survey responders such as Viola for regular tests that measure both physical and mental well-being. The doctor says with each step, researchers here are learning more about how the brain ages and why some people like Viola are faring so well. Whatever it is that keeps a person's mind sharp, Roscoe McGuire has got it.
Shirt. Brown. Oyster.
At age 97, he's another Leisure World resident who's beating the odds of old age. And in these memory drills at the UCI lab, he sometimes beats the clock.
Remember I told you, I don't want any help with it.
Roscoe wasn't a championship swimmer or a great athlete. He'll also tell you that his diet wasn't especially healthy and that he often enjoyed a good drink or two up until a few years ago. In fact, no one's more surprised at his longevity than he is.
What do you think, this is a trite question that people always ask at your age, how do you think you made it this far and this good shape?
I have no idea. Because I didn't expect it would be this long. My father was 55 when he died, and all his siblings were either that old or younger. My mother, I don't know what happened to her, her sister, but As I say, my mother lived to be 82, or almost 82.
But Roscoe knows that for whatever reason, he's been lucky, much luckier than many of his friends who've lost their memories and much more to Alzheimer's. So why not him? Who knows? But he hopes that being part of this study will help unlock the mystery of how minds like his survive the march of time without getting trampled.
Well, do you ever imagine what might have been?
Well, yes, I used to, of course, I lived my whole life in fantasy. I've done that all my life. And, that's everything. Maybe that's what kept me going. As soon as I didn't like, I fantasized something. So I was always a movie star.
The Legion World Survey didn't start out as a study in Alzheimer's. In fact, the questions were aimed at women who had taken estrogen in middle age. But after the survey was broadened, thousands of women and men responded, and scientists realized they had tapped into a motherlode of data on aging.
So that's two of our participants, and you can see they're pretty amazing. I will never be on the Jay Leno show, especially in a bathing suit. So this is the way we started the study. Actually, in 1981, some scientists at the University of Southern California wanted to study aging and estrogen and a variety of things. And they sent a 14-page questionnaire to every single person who lived in the city that is now called Laguna Woods. Because it's a retirement community, by the way, in the last few census, Laguna Woods is the oldest city in the United States. And they sent out about 18,000 questionnaires, and they got 14,000 of them back. That will only happen when you've got a mean age of 80-something. And the individuals who are in the study reflect the individuals who live there. They were primarily white because of the advanced age of the town. 2 out of every 3 people were women. Because by age 80, 2 out of every 3 people alive are women. And their mean age at the time in 1981 was 73 years. Now, 4 times subsequently, over the next 20 years, they were sent refresher sort of questionnaires, and they updated their information. And the information in these questionnaires was a variety of things that included some diet, some exercise questions, some medication questions, whether or not people took vitamin E tablets, things like that. So what we did after I arrived at University of California, Irvine, was we took those 14,000 people and we said on January 1st, 2003, if you were still alive and over the age of 90, you were going to become part of our study. So this study actually isn't just any 90-year-olds. You had to be in the original cohort, so we all also would have diet and lifestyle information from 20 or 25 years before that we could use to try to study. If you took vitamin E, for example, in 1981, did you live longer than if you didn't? Or if the women took estrogen, did they live longer than if they didn't? And that was approximately 2,000 individuals that we went after. Those individuals now reside in at least 35 states, so we have traveled all over the United States to see these individuals. And we successfully enrolled 83% of them, which is really, truly remarkable, and would only happen if you're over the age of 90, I'm sure. So to date, we've enrolled more than 1,600 people, making it probably the largest study in the world of its type. And the data I'm going to show you is what comes from those 1,600 individuals. We asked them to do an incredible amount of things. So we not only find out a lot about their medical history and their medications and their current lifestyle and diet, but we also give them a variety of tests, particularly about their memory, their language, their ability to think. You saw in the video they get neurological examinations. We ask for permission to talk to their family and friends and get information from the informants. And we ask them to give us their DNA and their cell lines. And in fact, next year we're probably going to be trying to make pluripotent stem cells from the cells of some of our participants. And then finally, when all is said and done, we ask for the ultimate donation, which is to let us study their brain so that we can better understand the things that we learned about them during life. And here's what the 1600 looked like. So they were fairly well educated, and 41% of them were college graduates or more. The majority were widowed. By age 90, out of every 4 people alive, 3 of them are now women. And honestly, this is something that somebody needs to figure out, because in every species where there's a difference in longevity, it is always the females that live longer than the males. It is one of the things that we clearly do better than the men, and yet nobody really knows why women should live longer than men, or female animals of any sort will live longer than the male. When we looked at their cognition, about a third of them were normal, about a third of them had dementia, and about a third of them were in between, where they had some kind of problems with their thinking or a little poor memory, but didn't qualify to have full-fledged dementia like Alzheimer's disease or that sort of illness. We asked them first. The first question we asked of this was, if you took vitamin C or had a diet high in vitamin C and supplements, did you live longer? And how many of you think vitamin C was associated with longer life? I've got a better question. How many of you take vitamin C? I had a feeling. Well, we started this whole project thinking that vitamin C and vitamin E as antioxidants would be particularly important for longevity. So part of our hypothesis was that in fact vitamin C and high diets with vitamin C and E would matter. And in fact, when we looked at the data for vitamin C, no dice. How many of you think vitamin A? Not quite as many, but pretty many. How many of you take vitamin A? Okay. So we had high hopes for that, and now my personal favorite was vitamin E. Vitamin E has had its heyday off and on over years, but it was particularly popular about 10 years ago. And we were fortunate to have information on which individuals took vitamin E pills and supplement. And how many of you take vitamin E? How many of you think it mattered? Now by now I was starting to get a little worried, because you're supposed to find out things with studies. Anyone think calcium mattered? And this is specifically for how long you live, not bone breaking or any other outcomes. And that's one of the things I do want to point out. I mean, this particular slide I'm showing you things associated with living long, but that doesn't mean these things aren't good for you, by the way. And that doesn't mean that some of these things might not be good for preventing other kinds of problems. But calcium, anyone think it was associated with longevity? Oh, yeah. Soft drinks. Anyone think soft drinks was associated?
Negatively.
Anyone think soft drinks was bad for longevity? Soft drinks didn't matter. Wasn't good, wasn't bad. Tea, green or black? And now I'm really starting to worry because, we were given like $12 million in 2003. And alcohol. How many people think alcohol was associated with longer life? What did somebody say? Yeah. So I have to tell you, I couldn't have picked these results better than if I had made them up. Alcohol was associated with longer life. Found in many studies. It's often called like the French paradox or something. But in fact, just like all those other studies in Europe and everything, we found that individuals who reported some alcohol intake, not necessarily a lot by the way, as little as 1 or 2 glasses a week was enough to generate the effect. And compared to individuals who did not have any alcohol intake, individuals who drank modest amounts of alcohol lived longer. Caffeine. Anyone thinks caffeine is good for you? Yeah. Anyone think caffeine's bad for you? Almost as many, huh? Caffeine turned out also to be good. Yeah, for those espresso groupies here, huh? Now, caffeine was U-shaped, is what we call it, which means that high amounts of caffeine or very low or no caffeine intake was not as good as moderate caffeine intake. And the caffeine intake that worked the best was between 200 and 400 milligrams, which many of you know is 2 to 3 cups of coffee, depending on the size of coffee, maybe 4 even. But we put all kinds of caffeine in here. So the caffeine that people get from chocolate's in there too. And the caffeine people get from tea, which has caffeine also, is in there also. But all forms of caffeine together, between 2 and 400 milligrams a day, was associated with longer life. Okay, body mass index. Body mass index is a way of describing whether or not someone is overweight or underweight. It takes height into account as well as your weight. And how many people think that being overweight was bad for longevity? And how many people think that being underweight was good for longevity? And how many people think being underweight was bad? And how many people think overweight was good? Well, I saw at least one lady there who absolutely got it right. And in fact, this I'm positive about, because the week we published our paper, actually in the American Journal of Epidemiology, the CDC came out with an even bigger study of about 500,000 people in the United States, and they got exactly the same result that we did. And their result was statistically significant because it was so big. And that is that individuals who were overweight in their 70s had a 3% reduction in mortality compared to normal weight people. And individuals who were underweight in their 70s and 80s had a 50% increase in mortality. It is definitely not good to be thin as you get older. I want to be clear, though, age matters. Age matters in so many ways, and this is one of the big ways, too. It was never good to be obese. So individuals in our study who were obese at age 21, even if they lost the weight, carried excess mortality for the rest of their lives. But what we found was that when you get to be in your 70s, 80s, and 90s, being underweight is terrible, and being overweight starts being at least not bad, if not actually good. So being overweight when you're 40 is not a good thing. But being overweight or maintaining your weight as you get into advanced age really is associated with longer life. Exercise? Anyone think exercise was bad for you? I was hoping. I'm a couch potato. Exercise exercise absolutely was good for you. 15 minutes a day, we found a longevity advantage. 30 minutes a day was even better, and 45 minutes a day, thank you God, was the best. And after that, it didn't matter. 3 hours was the same as 45 minutes. So for a long life, 45 minutes on average a day. It did not have to be done all at once, by the way. It could be 15 minutes of walking, followed by later in the day 15 minutes of stationary bike riding, followed by 15 minutes later in the day of, pulling up weeds in your garden. But on average, 45 minutes a day was associated with longer life. Leisure activities, like coming to hear this talk would go into that. Doing crossword puzzles would go into that. Any kind of meeting, social engagement, clubs, things like that would go into leisure activities. And how many people think it was good? It absolutely was. Anyone think it was bad? You can't raise your hand now. And it never leveled off. We took that out to 8 hours. 8 hours a day. So the individuals, for every additional hour that they spent engaged in activities, the longer their life was. So that's why being here is nothing else. You are going to add some time to your life, hopefully. Now, let's go back to this I showed you a minute ago. I said we really didn't know what to make of it because the data is all over the place after age 90 and 95. And so we took a look in our study. And the way you do that is we started with the individuals who were not demented in their 90s. And every year, we actually see these people twice a year, we established who developed dementia, and that's how we generate these lines. And when we did it, here's what happened. So the bad news is, the risk of dementia does continue to double with every 5 years of age, even after the age of 90. So you can see that our estimate for 90 to 94-year-olds is right in the middle of everyone's. But this is for 95 to 99-year-olds, and it doubles up to about 20% per year. And then over age 100, a staggering 40% per year. That means the individuals we're following who are over age 100, 40% of them every year develop dementia. So staggeringly high risk of developing dementia. And what is this from? We don't really know. And we don't know what the risk factors are either. So remember the things I showed you a minute ago? And these things, some of them were associated with longevity and some weren't, but the real important thing is not living long, it's living well. So we wanted to see if you took vitamin E supplementation in 1981, were you less likely to be demented? Or if you took vitamin C or had a diet high in vitamin C, were you less likely? How many want vitamin E? How many think vitamin E was good for dementia? I was really hoping. C? Any of the other favorites? Yeah, that was my real favorite too. So I'll make quick work of this one though. Oh man is right. We did not find any of these things to be related to the risk of developing dementia in your 90s. And some of these things, by the way, in younger individuals have been associated, especially things like the homocysteine levels, which is a marker of cerebrovascular disease or thyroid function in some studies. But in our hands, it didn't appear to be related to whether or not you developed dementia. So the next thing we thought was, well, you know, risk factors for vascular disease. That cause vascular diseases lead to heart attacks and strokes, and in younger people we've started realizing that maybe some of those things are related to your risk of dementia. And so we took a look at all of these. About half of the sample, for example, had hypertension, and 12% had heart disease or had heart attacks. A quarter of them almost had atrial fibrillation, a quarter of them had a stroke. Any favorites among these?
5%.
So, nothing was related to your subsequent risk of dementia except hypertension. How many people think hypertension was bad for you? Anyone want to go for it being good for you? Either you figured out my modus operandum or you've read something. That is absolutely right. What we found, and actually what several people have found, I think this is really important because this I'm also sure of is true, although I don't know why. But similar to gaining weight, with age, having a history of hypertension appears to be protective for the development of dementia. And this has been found not only in our study, but also several studies done in Europe, as well as Asia. So a very paradoxical finding. And you can see on our slides here, I'll point it out in a second, but not individuals who are pre-hypertensive, that is, their blood pressure, when we saw them in the clinic, was anywhere between 120 and 140 diastolic, and systolic, and 80 to 90 diastolic, those are pre-hypertensives, or hypertensive, those who were greater than 140 over 90. you can see that the risk of dementia decreased by 70% for the pre-hypertensives and by 80% for the individuals who had a high blood pressure when we saw them in the clinic. I think that some of what's going on here that is amazing is we've always assumed that whatever we learn, when we do a study, whatever we learn, like in Framingham, we study 30 and 40-year-olds to see their risk of whatever, heart attacks and things when they're 60 and 70. And we've always assumed that, in fact, is one of the studies that taught us that high blood pressure, high cholesterol, and things were associated with bad outcomes. And we just assume that that's the same for an 80 or a 90-year-old. And one of the main things I want to bring home today is that 90-year-olds are not just bad versions of younger people. So it is protective as you get older to have high blood pressure in regards to the development of dementia. And many of us around the world are trying to figure out exactly why that is. Some possibilities, for example, are just that, whatever is normal for a 90-year-old might not be the same thing as what's normal for a 50 or 60-year-old. It might be that when you have atherosclerosis and your blood vessels and your pipes and things are a little bit plugged up, that you need a higher blood pressure to get adequate blood to your brain. It might be that having a normal or low blood pressure is actually a marker of other diseases, and maybe those diseases are what's relevant. It might have nothing to do with blood pressure. It might be that the medications that we give people for blood pressure, for the treatment of blood pressure, actually is protecting them. So there's all different kinds of ways this can be interpreted, but right now, even though I'm sure of the finding, I really am not sure about why or how the mechanism for this protection is happening. Now, because all the traditional risk factors weren't relevant in the oldest old, we started taking a look at some new risk factors to see what is related to the development of dementia in these individuals. And here are a couple of very interesting ones that we're also pursuing. So you can take a little gadget like you see in the top photo there and put it on someone's finger. Probably most of you have had one of these in the hospital for a few minutes or something. And it tells us the amount of oxygen saturation in your blood. Normal people, most of you in this room, will have a saturation of 97, 99, 96. But if we looked at the individuals who had oxygen saturations below 93%, those individuals turned out to be more likely to have dementia. And so we're working right now on trying to figure out if maybe fixing oxygen saturation can contribute to improved outcomes. We also found out that people who walked slowly or had weak hand grips had much more likelihood of having dementia. And that's different from younger people, because a young Alzheimer patient, for the most part, walks just as fast or faster than I do. But as you get old, there appears to be a relationship between physical performance, walk speed, ability to sit up from a chair, as you saw one of our participants doing, or even hand grip, the power of a hand grip. So individuals who had the slowest walk speeds were almost 12 times more likely to have dementia. Individuals who were in the bottom quartile of hand grip were 5 times more likely to have dementia. And individuals who had a history of hypertension had a 30% reduction in their chances of having dementia. But what kind of dementia are they getting? So we've thus far enrolled 300 people into our autopsy cohort, and 185 of them have died and given us their brains. We get the brain very, very quickly, no matter where they are in the country, because we want to be able to do studies that need a very short post-mortem interval. And we look at the brain to see whether they were demented or not, what we can find in it. For the next little series I'm going to show you, individuals are divided into demented or not demented. They're about the same age, you can see. We gave them a test called the Mini Mental Status Examination, that's that MMSE there. And a perfect score on that test is 30. So the individuals you can see that we called not demented, they were getting 27 out of 30 on average correct, which is a great score, a normal score for any age. The individuals, however, that we were saying had problems with their thinking that qualified as dementia, they were only getting 11 on average. So there's a pretty big difference in the cognitive performance of these two groups, and that's one of the things I really wanted to make sure you noticed. There's a small difference in their brain weight. You can see that the individuals who were not demented had brains that were about 50 grams heavier than the individuals who were demented. And the first thing we did was take a look at the demented individuals, and 57% of them, the pathologist said, oh, this person has Alzheimer's disease. So that works. The only problem is 43%, very similar to the study I showed you by Crystal, 43% of them, he said, this is normal brain, normal elderly brain. Never mind that they can't recognize their daughter when they were alive, but as far as he was concerned, it was normal brain. So once again, we found a similar result to Dr. Crystal's. And then it got even better because we looked at the non-demented people, the individuals that I told you had really quite good cognition, better than mine in some cases. And guess what? Half of them also had Alzheimer's disease in their brain, and the pathologist thought that they should be demented, and the other half didn't. So Alzheimer's pathology at this age doesn't distinguish people who have memory problems and thinking problems from those who don't. And that's very different from younger ages. And here's an example of that. On the far left, in the two panels, you can see that's a not demented person, and you can see the brain looks pretty clean. Those brown spots are what we're looking at. They represent plaques in the top panel and tangles in the bottom panel, and those are the classic findings of Alzheimer's disease. And in the far right, you can see a patient that had dementia from the study, and you can see there are plenty of plaques in the top and there are plenty of tangles in the bottom. And in the middle, you can see a lady who did not have dementia, and she's got even more plaques and tangles than the demented person did, and yet managed to maintain normal cognition. So what is going on here? This is a slide that became real popular in the field in the past year or two. And it's a hypothetical model of the development of Alzheimer's disease. We don't know if this is true, and the shapes of all these things are not necessarily true, but it's what we're all trying to investigate right now. And in the red, the first line that you see on the left, that is labeled amyloid accumulation. So that is the plaques that I showed you in the past picture. And the hypothesis here is that you develop plaques, at least, a long time before you start having problems with your memory, which is shown by the first dotted line, vertical dotted line. And then after a while, when you start developing all these other things, the memory problem gets much worse, and we call you dementia, which is at the second dotted line. And all those other things are different kinds of pathology and brain tissue loss. But the idea would be you develop Alzheimer's plaques in your brain a long time, maybe 20 years, before you actually develop the dementia. And if that's the case, then maybe the normal people who have amyloid in their brain, they just died soon enough that they didn't ever express it, but if they'd lived longer, they would have. It's sort of like atherosclerosis. You know, you have a heart attack when you're 70 or 80 or whenever it is, but you start putting down the plaques and things in your vessels that lead to the heart attack when you're 30 or 40, and it just becomes apparent to us later. So that's the hypothesis about this, and exploring that hypothesis, we decided to use new technology. And the technology you might have read about in the newspaper last year is called PET, positron emission tomography, to image amyloid, which is the main thing that is made in the plaques. And florbetapir, which was given the brand name Amyvid, was approved last year by the Food and Drug Administration to be used in the clinic for the evaluation of whether or not people have amyloid plaques in their brain. So we got some of this compound and we gave it to about 13, 14 of our subjects. And the first thing we found was this. So all of those dots are an individual person, and the individuals on the right half of the screen are the individuals who have high amyloid uptake. And the individuals in the top left have low amyloid uptake. And if this was being used to diagnose Alzheimer's disease, then the physicians would say those on the high, to the right, have preclinical Alzheimer's disease. Those on the left don't have amyloid, so we don't think that they've got preclinical Alzheimer's disease. And what you can also see, because what I've graphed here, is that the people who have the high SUVRs, the high amyloid uptake, also have the lower scores. A perfect score on the axis is 100, and the individuals who had close to 100 actually had low uptake. The individuals with, so these individuals have no amyloid uptake, and they've got good scores up near 100. These individuals had high amyloid uptake, and their score, while still in the normal range, I think it's important to point out, a score of 80 or 85 can still be normal. It's borderline when you're talking about 80, but definitely that person on the far end who's got a 90, that's still a normal score. But you can see that the people with high amyloid uptake don't have as good of scores as the individuals with low. But I was more interested what's going to happen to them in the future, because when I looked at this with an autopsy, I can't follow people. But when I looked at it with amyloid imaging, then I can watch and see what happens over time. And my jaw dropped when this happened. So on the left, you can see the individuals that were considered, by the way the test is used, to have no or low amyloid uptake. And on the right, you can see the individuals who were considered to have high uptake. The individuals with low uptake over 18 months remained stable, with one exception, that one person who just who went down. And in fact, that person just died, so we're going to get a chance to look at her brain in the next month. The individuals with high amyloid uptake, though, they universally declined. I mean, they're bouncing around a little bit, but they're not doing well, and for the most part, every time we tested them, their score was worse than the 6 months before. A couple of them actually have developed dementia now, because you can see some of them are getting down into the 70 range, which is not normal. So it appeared that individuals with positive amyloid scans were at higher risk of developing dementia. So going back to this, I think that when you look at non-demented people who have AD pathology, the hypothesis we're working with now is that those individuals are at higher risk of developing disease and might be in a preclinical stage of Alzheimer's. But how about the people who are demented, that 43% over on the left, who are not demented, who are demented, and the pathologist said, oh, this is a normal brain, what's going on with them? And right now our best guess is that it turns out when you look at a brain of these individuals, they might be normal in the pathologist's eyes, but in fact they don't look like a 20, 30, or a 40-year-old brain. There are changes there. And the changes often can be fairly mild, and so to the pathologist it's, oh, this is a 95-year-old, so I saw a few little strokes, but they're microscopic and they couldn't possibly matter. Or, oh, I saw a little bit of scarring around the hippocampus, which is the part useful for memory. But, you know, I don't think that's a big deal. And in fact, probably it is a big deal. Because here's what happened when we looked. Even if the pathologies were very minimal or mild, if you add together the number of pathologies that we found in each brain, you can see that the chances of being demented go up dramatically. So individuals with 0 pathologies on the report still had a 41% chance of having dementia. One pathology raised it to 60% chance. But if you had 2 or 3 pathologies, your chances of having dementia were 93 and 100%. So we're starting to get the impression that multiple mild pathologies may account for a lot of the dementia in individuals in this age group. So, in summary, the number of people worldwide over the age of 90 and 100 is growing remarkably. It's going to be 4 times the number of people we currently have in the world. And unfortunately, the risk of dementia as well as other healthcare outcomes is extremely high in these individuals. And will be a huge public health issue if we don't find something to stem this tide. We've added 27 years of life in the last century. But a lot of that has been disabled life rather than abled life. So we need more research to understand the oldest old, the risk and protective factors, the things that contribute to dementia in this age group, because old people are not bad versions of younger people. And this came from my local newspaper. So thank you very much. It's really great. I more or less ended right on time, but I think if it's okay, can we have a few? Oh yeah, a few people want, huh? Okay, let's start in that corner.
In the list of protective or unprotected factors for Alzheimer's, you did not list exercise. You would have that in the protective factors for longevity, but not for the Alzheimer's or for dementia. There was a recent article in the New York Times that said that exercise protected the shrinkage of the brain, so that older people who over 70 who exercise regularly maintained the size of their brain better. So I want to know whether exercise matters for the prevention of dementia.
So the short answer is I don't know, but if you invite me back, I might be the. You know, there's a ton of interest right now in exercise, and exercise can work in so many ways that we know are true. I mean, obviously, cardiovascular health is probably good for all of these outcomes, but also exercise, it turns out, revs up something in the brain called BDNF, brain-derived neurotrophic factor. And BDNF encourages the brain to make new neurons and keep the ones that are there plump and things like that. If you take a mouse or a rat and you give them a wheel versus not giving them a wheel, the BDNF in the brain of the rat that gets to run is much, much, much higher. And there are other things that induce it too with exercise. In my heart, I believe exercise is important, especially in the oldest old for cognition. I'm not convinced it's important. But I have people in my clinic who unfortunately have developed Alzheimer's disease as young as their late 30s, 40s. I have, unfortunately, quite a large number of patients who are in their 40s or 50s even. I don't think exercise probably mattered for them, but especially with physical performance data, I'm leaning to the idea that exercise does matter in these individuals, and we're actually trying to devise an exercise program to prove it. The study you saw, what it did was just say that if we imaged your brain and we measured the volume, over the course of a period, it appeared that people who exercise didn't shrink quite at the same rate. And that's provocative, but it doesn't prove it yet. So, yeah. Yes?
How many of your 90-plus people are smokers or have been smokers?
Oh, has-been smokers was pretty high. I mean, in keeping with their generation, I mean, I think it was almost a third of them, at least at some point. Less than 5% are current smokers. I don't usually say this, but Jean-Louis Calmet, she smoked until she was 100 or 99 or something like that. And then, although her cognition was good when she died, her vision and her hearing wasn't, and apparently they made her stop because she almost set something on fire once. Some people just have the genes. It's interesting too because, I mean, obviously she had the genes of some sort. I mean, she not only lived to be 122, but she did it smoking, and her heirs did not daughter was quite sickly and died in her 30s. And her daughter had a child, a son who Jean-Louis raised, and he became a physician, and unfortunately he also died, I guess in his 30s or 40s in a motorcycle accident. So she leaves no DNA on the planet. Let's come over this way a little bit.
Could you comment on why you think your results with the risk of dementia over 90 were so much higher than the other studies you showed in that slide?
So the question was, why did my risk of dementia so much higher than the other studies? I think the main thing is that most of those other studies have very, very few people, say over the age of 85 or 90, and they tend to lump them all together and just report it as a risk for an 85 plus or a 90 plus. The advantage of having a large study is we had enough people to come up with a separate estimate for each 5 years. And I think that's why we see the doubling that also occurs before. Whereas other people were, maybe they had a few 100-year-olds and they just kind of lump them in with the 90-year-olds. Yes, ma'am.
You didn't mention lifestyle, attitude, diet very much. I'm curious what you think.
So we really don't have much data from this particular study that will allow us to look at lifestyle and diet, which is the question. What's the relationship of these? There is no doubt in my mind that diet matters. I'm absolutely convinced of this. But I think we've been making a mistake. I think that we want a magic bullet and it's not there. Everyone wants to take a vitamin E pill or a vitamin C pill. And they want it to be that one thing, vitamin E or C. I think Mother Nature understands diet a lot better, and she mixes things in food in a way that matters a lot more. And I think that even though doctors like to take credit for why we're living 27 years longer than we used to, and to some extent they can with antibiotics, a large amount of our increase in longevity is undoubtedly due to better nutrition. And I think that people who have studied diet in what they call dietary patterns, like the Mediterranean diet or things like that, that is, if your diet is not just high in vitamin E or C, but if it's high in fish oils, low in saturated fats from meat, has high in leafy vegetables and those kinds of things, then we do find a relationship with the development of dementia. So I think diet matters. I just think that the pills probably don't. Yeah. Come in.
You mentioned diet, exercise, and smoking, and so on, but you only passed briefly over the genetic input, the significance of genes. You just mentioned it a moment ago.
Yeah.
In weighing the two, the lifestyle, it's nature, nurture again, but how much would you weight genetic component as opposed to your lifestyle?
Yeah. So the question is, how much is it genes versus lifestyle to make you live long and live well? And everyone always talks about nature and nurture like you have to pick one of the two. And the reality is it doesn't matter what you're talking about, it's both always. And our genetic studies are in their infancy, so I can't yet tell you what about the genes of these individuals matter. But I do know, like all studies have shown, if you have long-lived parents, you are more likely to be long-lived, even if your parents had a completely different environment growing up than you do. There have been estimates about how much nature matters and how much nurture matters. I think it is probably hovering for pretty close to 50-50 in my view. If I was going to say one of them is less than 50, I think the genetics. I think the genetics are more likely to be, 35% or so of the variance, and that your lifestyle factors, whether you can control them or not. I mean, some of these are things you can control and some aren't. But I think that the exposures are at least 60%. And one of the things that, even in younger people we know, if you develop Alzheimer's disease, if you're an identical twin and you develop Alzheimer's disease, there is only about a 40 to 50% chance that your identical twin will also develop it. So that gives you a measure of when two people have, at least they're born with identical genes. The other thing is that even in twins where both of them develop it, often it can be as much as 14 years apart, and that's the exposure difference, I think. That says that one twin with the same genes got it 14 years later, probably because of something that happened after birth, you know. Whether it be something they controlled or not, I don't know, but, okay. The lady there?
Did you consider hours of sleep for 24?
Oh, the question is, do we consider hours of sleep? I am certain sleep matters, and actually we are starting to do some studies. In fact, we think that our oxygen finding may be partially related to sleep, and our blood pressure findings may be partially related to sleep. Because when you sleep often, your blood pressure goes down a lot. And I think that if you start out with normal blood pressure and your blood pressure goes down a lot during sleep, that might be a problem for getting enough nourishment to your brain, especially at night. Some studies have suggested that especially people who have sleep apnea at night have higher risks of developing cognitive decline and dementia. When we looked at total number of hours asleep reported either in 1981 or currently, though, we didn't find a relationship with just how long you sleep. So I think the quality of your sleep and what goes on is important. Let's try. Over here.
I am a month short of being 99 years old.
I've got a consent form you can sign. I think that supplementation contributes. And you have to eat my feeling in medical school was, how could you possibly get all those vitamins and minerals that I'm learning are necessary for reactions, you know, just from a meal or two, which is all I usually eat a day. And I think that especially if you're not getting your nutrients from your dietary intake, you really probably do need to consider supplementation. But afterwards, maybe you can, I'll write it down because I want to go look up what you're taking. 25 years, so she started with 75, right? Excellent. Dr. Whitaker's, we're putting in a plug for him. Yes?
You mentioned that there are other studies that collect the data on older women who are taking estrogen replacement therapy. Have you or anyone else found conclusions about that?
So, oh my God, I guess I took it out of this talk because the question is, what do we know about estrogen? And I didn't even like OBGYN or anything, so I don't know why every time I turn around I'm publishing a paper about estrogen. And every time I turn around, it is a paper that says something good. So, in fact, in our study, one of the things that definitely was associated with longevity was estrogen use, perimenopausal estrogen use. The women who reported that they used it when they were in their 50s or 60s or whenever they had menopause lived longer than the individuals that didn't. And this has been in a bazillion studies. I mean, the pendulum for estrogen sort of swings back and forth. And I think part of the problem is that it's not the same if you take estrogen perimenopausally as if you do what we tried to do in some of the clinical trials like Women's Health Initiative, where we gave women in their 70s and 80s estrogen. So at least in this study, the women who reported estrogen supplementation around menopause actually lived longer than women who didn't. Now, when it came to dementia, the results are a little not so clear, so I'm not convinced, but we're working on that one too. Yes, sir.
I was wondering, you mentioned, I think the study's funding is around $12 million. That was two questions. One, where's the money coming from? Who's funding it? And two, you also mentioned that people are living longer, but not necessarily better. And I have seen other studies that show that mobility and things like that are actually going down in the later years. So I was wondering if you would comment on that a little bit.
Yeah. So the first question was, where's the money coming from? And all the money that has been used in this study, with the exception of those 12 Amivid scans that I showed you, has come from your tax dollars through the National Institutes of Health. All of my funding is from the National Institute on Aging. And the second question is, what's going on that mobility is actually getting worse? And I think some of this might be that, at the turn of the century, life expectancy was actually 49 years for women and 47 for men. That's in the early 1900s. At the turn of the 19th. Well, in 1900, in 1900, life expectancy was actually in the late 40s, and then of course by 2000, life expectancy was then in the high 70s, and so that's where I'm getting sort of the 26 years. But part of it also was when you became disabled or sick in the past, you tended not to live so long. And now we're keeping people alive with a lot of chronic illnesses and disabilities. And if you have one of those, we call it, disabled life. So a significant part of that 27 years has been, for many people, not an extension of good life, but an extension of disabled life. I don't think that mobility itself is going down if you consider it age-specific. I mean, I think that individuals who are 60 now are walking as well, basically, as individuals who were 60 100 years ago. The only difference being that we've kept some people alive to reach age 60 with disabilities that didn't happen before. So, yeah, let's try over this corner. My gosh, there's more. I should have just done questions. The question was, any research correlation on vitamin D? Unfortunately, vitamin D wasn't in vogue in '81, so they didn't ask them, and I didn't have that kind of measurements. I kind of have a pet peeve about vitamin D because all of a sudden it went from nobody taking it to, how many people in this room have been told by your doctor you have low vitamin D levels? Yeah, as many as in California, so sunshine doesn't seem to matter. And how many of you did your doctors put you on vitamin D? Yeah, as many as California too, and my parents in Kentucky and my aunts in Texas. The vitamin D data is not terribly convincing to me. I don't understand even what a normal level should be. And you would think that if vitamin D really mattered that much, you should be able to see a gradient. That is, that places with a lot of sunshine and hence people should have higher levels, should be have less Alzheimer's, live longer, whatever it is. And in general, that doesn't clearly appear to be the case. So I think, although vitamin D might be important, I'm not aware of convincing data yet for either of these things in aging. Yes, sir.
I'm curious about the value of having a spouse, both with respect to having longevity and the development of it.
Oh, I love that question. So this is well established. Married men live longer than single men. Married women do not live as long as single women. So we can do 2 or 3 more questions, and I'll try and answer them briefly and at least get in 3. But seriously, even though it's pretty funny, it's true. Now, the relationship...
Some men want to die young.
Now, the relationship with dementia is less clear, particularly since, I mean, it's very hard to tell when you're dealing in your 90s and you've got so many widowers. Having said that, having a partner of whatever sort, I do think is similar to the leisure activities data I showed you. I think it increases socialization, well-being, attitude, and all those things that matter, probably for both sexes. Back here.
What's your opinion on alternative day fasting for reducing dementia?
I'm sorry, say again?
What is your opinion on alternative day fasting for reducing dementia?
Oh boy, alternative day fasting for reducing dementia? Okay, that's the question. So most people usually ask about fasting for making you live longer, because I absolutely, unequivocally can make a rat live longer if I basically starve them. And that is well established. Calorie reduction or restriction to a severe level makes rats live longer. And a few years ago was the first evidence suggesting that you could do the same thing in a primate model. Although I really want to point out that in that data, what they said is that the individuals, the primates that they starved essentially, had fewer deaths due to what they call age-associated diseases, like diabetes and things like that. But it turns out that the animals that they starved actually had virtually the same death rate as the animals they didn't, because they were more likely to die in other things, like when they did surgery on them or something like that. Calorie restriction in animals may prolong life. I'm not convinced yet that it does it in humans, especially when I see data that says if you're overweight, you do better than if you're normal weight, and certainly better than if you're underweight. And I've never seen any data suggesting it's useful for dementia.
Any association between diabetes and dementia?
Association between diabetes and dementia is the question. So you've probably heard some people calling Alzheimer's disease type 3 diabetes and some other terms like that that maybe in part bring up the question. I mean, don't take those too literally. What people are really saying sort of is that sort of the failure of neurons to use sugar and things like that is contributing. The main association that's been found between diabetes and any dementia has always been through vascular risk factors and strokes and microstrokes, because diabetics do have higher rates of disease. Having said that, at least 5% of our sample are diabetics, and it is not in this sample associated with dementia. So I think diabetes is a largely misunderstood disease, and maybe with better control of blood sugar and things as we're now doing, maybe that's partly obscuring any relationship that might be there. But if there is a relationship, I think it's more likely through vascular than Alzheimer's mechanisms. Yes, ma'am.
You said that high blood pressure is good.
For the outcome of dementia, right? It might be bad for a heart attack or something else, which is also work we're doing. No. The question was, high blood pressure appears to be good. What's the maximum safe blood pressure? And the problem with this kind of study is it's what we call an observational study. So I can't quite control what people's blood pressures are or anything. I just get to watch them. What I would say, just as a physician, is I don't think sky-high blood pressures are good. I don't think that 200 over 110 is ever good. But I think that blood pressures that are above what the American Heart Association is telling us is good for 50-year-olds is what's good. So my guesstimate, if I had to say an optimal blood pressure as we observe it in the study, it would be something that's, say, between 150 to 170, 75 as a systolic upper limit, and 90 to 100 for the diastolic. I think you don't want it higher than that. But the American Heart Association considers that too high, at least for younger people. Right? One more. Oh God, you have to pick out who it is. Just point to them. Yeah. Over there.
A relatively high proportion of it seemed like of people in your sample were fairly well educated. So were you able to look at education and its relationship to dementia and perhaps any other intermediate factors that affect education and dementia?
So the question is, how is education related to dementia? And actually, my mentor, Robert Katzman, was the first one to describe there is definitely a relationship between education and dementia. And if you are highly educated, your risk of getting dementia is actually lower. Which is not to say that Nobel laureates, presidents, all sorts of people get it, unfortunately. In the first study that he described it in was in China, where fully a third of the women had never held a pencil in their hand because they'd never been to school. So to find an education effect, it helps to have a nice big spread. And as you pointed out, the range of education in this white population in Southern California was really quite narrow. So in general, every time we've looked at the relationship of education and anything in the sample, we don't detect anything. So that's different from saying I don't think it matters. Education is definitely related to the risk of dementia. But once you've got people, some of which did college, some of which did a master's, and some of which, you just don't have enough of a range to really show it. Okay, thank you very, very much.
Join us in the lobby.